Hi there, this is our second week, and this week the PPE we need is our lab coat and gloves. For goggles, it's up to you. If you feel like you might get a chance to splash DAPI into your eye, you can wear it, otherwise you don't have to. Alright, get ready and go. First one, we need our slide, which is this guy here. We're gonna put our brain samples on top of here. And then we'll need a cover slip, which is this guy here. I'm gonna put this guy on top of our sample to protect our sample. And then we'll need a 24 wall plate. We're gonna put brain into these little walls here, and we're gonna use different solution to incubate them. Then we need the PBS, and then we need the brush. We're gonna move the brain slice with that, and then we need a micropipette. This one is 20 to 200 microliter, which means the capacity for this one is up to 200, and the minimum amount is 20 microliter. And then we need the tips for this guy here, and of course we need some DAPI solution, which is in here, and then we need a mounting medium. This is different than PBS, so this guy is called a fluorophore mount. So mount means mounting medium, and this guy is very sticky. If you get that thing on your hand, you're gonna get like very sticky. It's not very pleasant. Okay, and then we need a petri dish to put all these brain slices in here so we can sort them out. Okay, let's go ahead and start. So first one I need to do is label my 24 wall plate. I already labeled here. And then if you wonder if you open the lid and then they get moved like this way, then your label is gonna be off, right? But do you see there are notch here? So the lid can only go to this in certain way, so you don't have to worry about that. As long as you can close the lid, label will match to the plate. So next one, I need to fill up the plate with different solutions. Okay, let's take a look how to use the micropipetter. So this guy has 200 microliter capacity, so I said 200. So if I need a 400 microliter of PBS, I just need to do twice, right? Okay, and then how to use this guy. So if you push one time, it's going to get rid of the air, and then you release, the solution will get into the tip. And then now when you're ready to transfer the solution, there's two stops. So the first stop is to get rid of solution. The second stop is to get rid of the little amount left in the tip. However, in this lab, I don't recommend you to go to the second stop. Why is that? Because in this lab, the volume accuracy is not really matters. If transfer 200, 190 microliter, doesn't matter. But if you go to the second stop, you are going to generate a lot of bubbles into the solution, and that's something that we don't want. So don't use the second stop of your micropipetters, and that only in this lab. Later on, if your accuracy matters, you have to go to the second stop. Okay, let's transfer PBS into our solution, into our walls. So I set my micropipetter to 200, and I just need to do twice. So that's my first stop. I didn't go to the second stop, and you see there's a very little amount of solution left in there, which is not a big deal. Okay, if you go second stop, see the solution get out. For this lab, we don't need that, so twice. So next one, I need to transfer 300 microliter of DAPI into my 24 wall plate. So I need to set up my micropipetter into 150. I just need to do twice. Before we go, I need to change the tip, because the tip is PBS. I don't want to contaminate my DAPI solution with anything else, right? And I have a waste container, so I'm gonna put my tip into my waste container, and that's the fun part. If you push this guy here, the tip will go into the waste container. Ding. Okay. All right. And then I need a new tip. I put a new tip on. And this is my DAPI solution labeled with D, so you always want to read labels. Okay, now I'm going to open that, and then I want to transfer 300 into my wall. So that's my DAPI. That's 150. And another 150. Okay, now let's transfer the bring slice into our plate. So this is the bring slice in here, and that's in storage medium. And this storage medium is kind of like sticky, so we want to get rid of that. That's why we go the PBS wash. The PBS will get rid of storage solution. So how do you transfer that? You can just dump all the bring slice all together with the solutions in here. Whoosh, like that. Okay, and then you need to check if there are 3 bring slices in here. Okay. All right, and that thing stays here for 2 minutes. Right, now it's 2 minutes. I need to transfer the bring slice into this petri dish because we need to sort them out, right? So first thing, we need to put some PBS in this petri dish. So because we don't need exactly volume, I just need roughly 15 ml of PBS here. So this one, I have labels in here, so that's that's 35 ml. So I just need to pour that thing into 20, so that's 15 ml, right? Okay, let's pour that. That's roughly about it. This one looks like Avadora because that thing has eyes in there. See that eye look structure, the darker part? That's a hippocampus. So this is the most posterior one. And this one looks like the Bart Simpson's mom because this one is the hair and you have a little bit facing here. And this one, you can say that looks like Bart Simpson's father one, or that looks like a Kung Fu Panda with the cheek in there. And based on that, this one is going to be the first one, most anterior one, and then the middle one, and this one's the most posterior one, right? Okay, now we need to transfer that into our slide. And before we transfer that, we need to put some PBS on there, because if you don't, the bring slice is not going to attach to glass. So we're going to use a transfer pipette. Again, the volume is not important. This is the PBS that we used earlier, so I just need to take some PBS in here and put 3 drops. So I can get rid of them in here, and just be careful, don't get any bubbles in, because the bring slice don't like bubbles. The first one that we want to transfer is this guy, right? Because that's most anterior one, and because you see the dot there, that's anterior commissure, so that one's the ventral side, and then the Simpsons mom's head hair is the dorsal side, right? So we want the ventral side close to us, and then the dorsal side away from us. So I'm going to transfer that thing. Okay, now I need to use a Kim wipe tissue. We'll fold it into a corner, so you can use that to absorb waters. But before you do that, you want to put your brush here, otherwise the bring slice won't go with you. Adjust that a little bit, so make sure that the bring slice is facing the way that you want. I'm very OCD, so I want to get water away as much as possible, but it's not necessary. Okay, the first one, mission accomplished. Okay, now let's get ready to mount this tissue. So we need to put some mounting medium in there, right? So how do you put mounting medium in here? This is a sticky solution. You don't want to use your hand, your fingers. You want to use this tip, and just the tip is good enough. You don't need a micropipetter. So you want to think this tip is a spoon. Okay, so how do you do that? You want to spoon it up and then drop it. Okay, I'm going to show you how do I do that. So if that is a spoon, you dip that, and you see there's some liquid coming along, and then you tilt your tip, and that thing will drop onto your slide. There we go. And next one. Yeah, cool. Okay, so you want to throw that away in our waste container. Okay, and the last thing, we need to put the cover slip on that. So here's the cover slip. If you drop the cover slip on here, you are going to generate a lot of bubbles. So you don't want to do that. So you want to lower one edge first, and then lower it down slowly. So that's when we're going to see the cover slip is touching the mounting medium, and you can go all lower. There we go. There's some area that don't have a mounting medium, which is okay, because as long as our sample is covered by the mounting medium, we're good. Okay, now let's take a look at the microscope. So first thing, you want to put your sample onto your stage. So there's a clip, you slide that thing in, and then you need to turn on the light, the fluorescent light. So because this is a DAPI, so we need the violet light, which is V here, and then we need to crank up the intensity to 100%. If you use 10%, the light is going to be very dim. And then why do we use violet? Because DAPI has the excitation light as violet, right? If you use other color, you are not going to see any result. And then of course, you don't need the bright fields. You want to make sure that's turned into 0, and you need to have this curtain cover your bright field light, otherwise you see the lens there is going to have some reflect light from the environment go to your sample, so you want to cover that thing up. Now let's turn on the fluorescent light. So if I turn that to W, there is a white light coming, and then R, it's the red light. See the red light? The light is coming this way, it's downward. Okay, and then this is blue, this is green, and then the cyan, and then the violet. Okay, so this is the light that we need. Okay, so now the light is coming this way, and then when hit the sample, is going to be bounced back to here. And then if you turn your bino photo into bino, which is push in. If you push that out, that's going to go photo, going to go to your camera. So you want to first, when you start, you want to push that in, start bino, so you can see samples from this eyepiece. Then we have to make sure that our slice is right in the center of the light, so we can see that under the microscope. If that's off the center, we're not going to see anything. Okay, how do we adjust the lens? You will always want to start with this 4x lens, right? You don't want to use others when you start, so always start with 4x lens. And then you want to crank up the stage into the highest position. So this is the highest position. And when you're looking for samples, you are going to lower that down. Now let's take a look of our samples. So this is the image that you see when the stage is the topest position. Now I'm going to lower my stage. And you start to see structures, okay? And I'm going to find adjust side to find the best focal plane, okay? Okay, let's take a look at the image that I took under the microscope. So I totally took 12 image for the first brain slice, and because each of the image under the microscope, even that's under 4x objective lens, you can still only see a small part of that. So in order to see the whole structure, I have to put these 12 images back so you can have a rough idea about the bigger picture of this image. Now let's take a look at specific structures. First one is the corpus callosum. And because this slice is too anterior, so we don't have any corpus callosum on this slice, right? So we have to go to the second slide to see the corpus callosum. So the corpus callosum is this white color guy here, right? And if you transfer that to the DAPI stain slice, it's this guy here. And this guy, so corpus callosum. And next one, the midline. So we got midline here, and this is the midline. And on this one, you have midline here too. This is not a anatomical structure, but that helps you to orientate yourself on the brain slice. And next one is the cortex. So you can see on all these three slices, you have cortex. The cortex of this slice is pretty thick. It's pretty much from here, go all the way to the center. So this whole area is cortex. And for the second slice, the cortex goes from here to the boundary of your corpus callosum. So that's the cortex. And you can see from this slice, the cortex is gray matter and corpus callosum is a white matter here, right? And next one is the same here. The corpus callosum on here is thinner than the one you see on the anterior slice, right? But you can still see a little bit different color stainings because the nuclei density is different in corpus callosum and cortex staining here. And talking about cortex, let's take a look at the layers. Actually, we can see layers. There are 6 layers of a cortex. So you can see there is a very, very blue layer, very thin. That layer is not the cortex. That is the meninges. You probably remember there are 3-layer meninges. We cannot differentiate the 3-layer meninges in here. Okay, and then you see a darker color layer, this layer where my hand is brushing. That is the first layer of your cortex. And roughly in some region, you can see some layers. But on this part, you cannot see other layers other than the first layer, right? And I want to ask you why the color in first layer is darker. Is that because the density of the nuclei in first layer is lower, or the density of the nuclei is higher? The answer to that is the darker the color, that means there's a less DAPI stain. If there is less DAPI stain, that's going to be pretty dark, right? So there's no DAPI stain in the background, so it's the darkest. And this one is darker, that means there's less positive DAPI staining, which means there's less nuclei in here, in the first layer of the cortex. Next one, hippocampus. So we cannot see the hippocampus on the second slice. The first slice, we can't see them either. We have to see it on the third one, right? So this is the hippocampus. You can see more detail after the DAPI staining. And there are different regions of hippocampus. There is no distinct boundaries, but it's roughly. So see, one region is somewhere in here. and then there's region here, CA2 roughly, and then CA3, and then this peak here is dentate gyrus. Okay, now let's take a look at lateral ventricles. So you can see a little bit of lateral ventricle on here, which is this little long structure. And zoom in. Okay, so you probably think lateral ventricle should be a hollow structure with the CSF in there. However, you have some cells in here, right? And you think what kind of cell they are? They are actually the blood vessel cells. So you remember in the first week when we dissecting the brain, I dig out some blood vessels in the lateral ventricle. You remember that? Okay, let's take a look at lateral ventricle on this slide. So this is the lateral ventricle that we find on this slide. It's a very long structure. And the stainings that you see here is the blood vessel and the linings of the lateral ventricle. Okay, let's talk about the ACO, the anterior commissure olfactory limb. Okay, so on this slide, we have the ACO here and here, because the anterior commissure olfactory limb, that's white matter, right? So you see that's white in here, and you see the color is a little bit different than other part of the tissue. So that's ACO on this slide. You also have ACO on this one, this little white dot there, that's the ACO, right, underneath the lateral ventricle. And on this slide, you do have white matter in here, but that's not ACO, that's some other structures. Right, because when you see hippocampus in here, you know the ACO is not present. Okay, next one, nucleus accumbens. So nucleus accumbens is part of deep brain nuclei. They are also called basal ganglia. You can find them in the anterior slice. So on this one, the nucleus accumbens is roughly around the ACO. We don't have a distinct boundary, but that's roughly where the nucleus accumbens is. And next one is caudal putamen. Caudal putamen is also part of the deep brain nuclei, and that is right underneath the corpus callosum and laterally to the lateral ventricle. So that big chunk part here. Okay, let's take a look at the structures under higher magnification. First one is corpus callosum. So this is the corpus callosum under 4-axis objective lens. You can see a bigger view, but you cannot tell each different nuclei from each other, right? Okay, and then if you switch to 10x objective lens, this is the whole view that you see from the 10x objective lens. So it's a smaller, you see smaller view with higher objective lens. However, you can see the nuclei a little bit better. You can tell this one from the adjacent nuclei, right? And I want you to notice, did you see there's a kind of like a pattern of the nuclei follows in the corpus callosum? See, they kind of line up. So there's a lineup, 305, they line up. Underneath the corpus callosum, that is the caudal putamen, is that right? You don't have this pattern. And above the corpus callosum, that's a cortex, you don't have this pattern either. Am I right? Okay, let's go a little bit higher magnification. So this is the whole view of 10x, and then if you switch to 20x, you only get to see this part, and you see a little bit better image of these nuclei. See, they line up, line up, and then we go to 40 objective lens. So this square is the thing that you can see under 40x objective lens. See, they kind of like line up together, and you see there is nuclei, and actually you can see the nuclei has some dot in there. See the dot in there? I want you to think about what these dots are. Okay, next one. The dentate gyrus. So this is the 4x objective lens of dentate gyrus. This is the whole image. And then if we switch to 10x, we are only going to see this part of the dentate gyrus. Okay, and we need to take a look at the granule cell layer of the dentate gyrus. You see this really blue area there, that is granule cell. So there are there are a lot of cells here, which means the cell density here is really high, which means the nuclei density here is really high, which means you have a lot of DAPI staining here. So that's why this part is very blue. So the bluer you see, the higher nuclei density you have, right? Okay, so this whole view is 20x, and then you are going to see this view under 40x. So that's how it looks like under 40x. So you can see there's a big nuclei here, there's a nuclei here, and then you also see this dot inside the nuclei. Think about what these dots are. Okay, next one, we need to see the CA1 layer, and particularly we need to see the pyramidal cell layer. So this line here, that is a pyramidal cell layer. So this is a 4x objective lens, and if you switch to 10x, you are only going to see this little square here. That's 10x objective lens, so that's the 10x. And if you switch to higher magnification, you see inside this box that become this whole view here, and you pick up this view and switch to 40 objective length. So that is how pyramidal cell layer in CA1 looks like under 40x. You can see there's nuclei, there are dots in there. They looks very similar to the nuclei in granular cell layer. Is that right? Other than the granular cell layer probably have more nuclei compared with pyramidal layer, but in terms of the shape of nuclei, they don't differentiate much, right? See this nuclei in granular cell, and this one is a pyramidal cell. It looks pretty close. Okay, next one is a lateral ventricle. So I want you to take a look at under 10x. We already take a look at under 4x objective lens, right? This is a 4x, and if you zoom in, you will see there's a smaller part that's pretty dark. That is the hollow part of the lateral ventricle. This is the brain slice that we took the image from. And if you take the lateral ventricle from this image, you see there is a hole here. Then you're going to see a little bit bigger, darker region on the lateral ventricle. And if you take a lateral ventricle image from this slice, you see there's a lot bigger hole in here. That's your lateral ventricle is going to look like under 4x objective lens. There's a big hole here, and then the 10x here. You don't see any blood vessel in here because the blood vessel only take a small proportion of volume of the lateral ventricle. So the majority part is pretty much hollow because they were filled up with CSF, and you don't have any cell in CSF. So you don't have any nuclei, which means you don't have any stain after DAPI stain. I want you to know that the color you see on the monitor is pseudo color. What's that mean? That means that color can be easily changed. If I go to the gear symbol, and then I can simply just change the red channel, green channel, or blue channel. For example, if I move the red channel up, my color become red, right? If I change the blue channel, my color become more blue. Our microscope, the green channel is locked, so we cannot change the green channel. However, we can just easily change the color by changing the red and blue channel here, right? So how do you know that which color you're supposed to use for your image? Usually, the color that you put in here should match in the color that you see under the eyepieces. Usually, the first time when you look at your image on your camera, the color is going to be way off. So there is a shortcut. The camera can do a white balance for you. So if you click white balance and check it, it's going to do a calculation of what color is the closest color you can get. For this situation, it's not ideal. It's blue, but it's not the best. So in this situation, you'll have to manually change the color, make it a little bit more blue. So that will look like DAPI. And if your image is too bright, you can lower the gain. This is a computer calculation of image. You can make your image darker or make your image brighter. And the first time when you're taking photo under the camera, you can do an auto exposure. if you check this guy here, it's going to calculate the exposure time and then gain together, find which is the best for you. Both of these will change the brightness of your color. In this situation, we have 39 as our exposure time. If you increase that, it's going to make your image brighter. So increase that more. It's very bright, right? You want to lower that. The same thing, if you increase gain, it's going to make that brighter too. The difference between the exposure and gain is the exposure is by changing the shutter, you are controlling how much light going into the camera. And the gain is the camera calculating the image. So gain is the artificial one. And there's other settings, the saturation, gamma, brightness, sharpness, all these things are for advanced image setting.